Okay, guys, that gets us through understanding something about the regulation of the TCA cycle and that committed step of pyruvate dehydrogenase going into that acetyl-CoA pool, which we need to run the TCA cycle. And we've sort of crept back up this hierarchy of relatively simple feedback inhibition to allosteric inhibition to now the cyclic cascade. Now we're going to put some of those ideas together in some additional ways in terms of also thinking about the overall pathway design in terms of regulation of glycolysis, right? And remember, regulation of glycolysis is going to involve regulation of symmetric and inverted regulation of gluconeogenesis. We'll come back to that a little bit as we get to the end of this discussion, right? So if you think again about glycolysis, here's that basic pathway again, right? We're going to take glucose, we're going to run it through these intermediates and these steps we've talked about all the way down to pyruvate, which is that key step that we had to regulate. We just talked about it, pyruvate dehydrogenase. Now the regulation, if you recall, I've hinted at this a few times, in glycolysis exists at only a couple of places, really, right here at the beginning of the pathway, right? And at the very end of the pathway, right? And that, again, if you think about the pathway design, the way we've gone through it, that makes some sense. Because remember, for the purposes of running glycolysis and the advantage of being able to reverse this guy into gluconeogenesis, the central trunk of this pathway, those central 6 or 7 steps or so, are all operating relatively close to equilibrium or easy to push around. And there's essentially zero regulation going on in the core of glycolysis. It's all at the beginning and at the end. And by flipping those switches, you can run this thing in opposite directions. So thinking glycolytically, our regulation at the top is really in 2 places. Depends a little bit on the tissue. It's at the level of hexokinase or glucokinase, depending on your tissue. The biggie, though, at the top of the pathway, pretty much universally, is PFK, phosphofructokinase, right? Now, the way in which this enzyme is regulated is very tissue dependent. We're mostly going to be focused at a sort of a liver tissue level because of some of the additional layers of regulation that come in physiologically for PFK in liver tissue. But some level of this regulation also exists in muscle tissue and in other tissues, right? But there's a specialized element here we're going to consider in a little bit. At the end of the pathway, the primary point of glycolytic regulation focuses on pyruvate kinase, right? And some of this, again, I've hinted at, you've seen some pieces of it, and a lot of this kind of makes some sense, right? You want to control the forward pathway, right? So you then control the beginning and the end. And the things that control it, again, make some sense, right? So pyruvate kinase is sort of feedback inhibited, not directly by pyruvate so much, but by the next major pool, which is acetyl-CoA. So if the acetyl-CoA pools build up, you get a feedback, and it's an allosteric response because it's not a classic product inhibition. You get an allosteric response. This is a pretty sophisticatedly built and regulated enzyme, but sensing the acetyl-CoA pool. Turns out, for reasons that are very important, we'll talk about in a minute, the fatty acid pool, which also through beta oxidation, as you'll see when you see talked in Dr. Carr's lectures, also feeds the acetyl-CoA. Fatty acids will also regulate pyruvate kinase. Then, of course, the feedback inhibition, there's a feedback inhibition that makes a lot of sense. It's a direct product feedback inhibition with pyruvate kinase, and that's ATP. Production of ATP feeds right back in, classic product inhibition feedback that shuts down pyruvate kinase, and the ATP pools build up, right? There's another one that we'll talk about here in a second that comes from fructose 1,6-bisphosphate. It's actually an activator. It's an allosteric activator of pyruvate kinase. Okay. All right. So let's take a look at pyruvate kinase in a little bit more detail. I showed you some of those basic feedbacks, but let's look at it in a little bit more detail because there's an additional layer, just like we saw with pyruvate dehydrogenase complex. This enzyme is inhibited or controlled both at the level of direct feedbacks, allosteric effectors, and then a cyclic cascade, right? Okay. So here's pyruvate kinase. Remember the basic reaction, phosphoenolpyruvate, ADP. We're going to make pyruvate and ATP. ATP, as we just talked about, is a classic feedback inhibitor, product level feedback inhibitor. Turns out alanine is also, for other reasons, a negative regulator, negative allosteric regulator of the pyruvate kinase reaction. At the same time, I pointed to that arrow on that previous pathway of this fructose 1,6-bisphosphate, right? is an activator. It's an allosteric activator of pyruvate kinase. Look at that. Well, why would that be, right? I mean, fructose 1,6-bisphosphate, it's not a reactant. It's not the immediate. Why would you do that, right? This is an example of something that's, of course, known as feed-forward activation. And again, if you think about the pathway design for glycolysis, this makes a lot of sense. If PFK, phosphofructokinase, is active and running, and it's trying to push carbon equivalents down the glycolytic pathway, because of how close all those intervening steps are to equilibrium, how many of them are even biased against glycolytic flux, right? In order for this pathway to run glycolytically, you gotta have the drain at the bottom of the pathway full open, right? Or you're not gonna run glycolytic flux. It's just gonna all back up. So the product of that previous committed step, which is the PFK step, right? Remember, PFK is that committed step at the top. The product of its reaction, fructose 1,6-bisphosphate, feeds forward as an allosteric effector that regulates pyruvate kinase and cranks its activity up. And that makes a lot of sense. You want to keep those two committed steps sort of communicating with each other. So there's more coming in here. Well, you better open up the drain at the bottom, or we're going to, like, you know, flood the turbine and blow up the powerhouse, right? Okay, my analogy from last time. Okay, so that makes a lot of sense, right? So it turns out pyruvate kinase is also a very elaborately regulated at the level of a cyclic cascade, right? So pyruvate kinase, especially in the liver isoform of this enzyme, I'm primarily talking about here, when it's not phosphorylated, it's more active, right? And then there's a kinase that will take ATP, phosphorylate it, and turn it off, right? And a phosphatase then that will clip that phosphate off and return it back. So here's our classic sort of phosphate dependent cascade, and there's a kinase, as I said, that comes in here, and a phosphatase that comes in here. And in the case of the liver isoform of pyruvate kinase, what's controlling that cascade is the blood glucose levels, right? And we're going to see how that circuit in part is put together here in just a second when we talk about PFK, because we got to layer that enzyme on top of this as well. All right, so pyruvate kinase here at the bottom, elaborately regulated allosteric classic feedback, and also a cyclic cascade through a post-translational modification. All right, let's talk about the top of the pathway, PFK. Remember, our other committed step, really, other than a bit up here from hexokinase, glucokinase, is the PFK reaction. In this case, the feedbacks we've got are allosteric. There is somewhat less, or there isn't really a lot of feedback inhibition from its immediate product, fructose 1,6-bisphosphate, because it gets drained so fast when you're running glycolytically. There isn't much of an opportunity, if that pathway is really running, for this to feed back, right? That pool has to be kept pretty low in order to keep the glycolytic flux going the right way. But it turns out two downstream products of the pathway do feed back allosterically. One makes a lot of sense. ATP, of course, is a negative allosteric effector, feeds back and shuts PFK down, as does a downstream product that might make a lot of sense if you're thinking about not wanting to run this thing too much because you got plenty of ATP, and that would be citrate. Remember, the citrate's that committed product, or that product of the committed step TCA cycle. If you got a lot of citrate around, you don't necessarily need to be running glycolysis to make ATP. You got a lot of citrate, just let the TCA cycle do its thing. So the citrate pool is sensed by PFK as a feedback regulator of flux coming down glycolysis. Now, at the same time, you have some activators, right? ADP is a classic allosteric activator of PFK. ADP levels build up, ATP levels are dropping off. These guys are in opposition, cranks the enzyme up just sort of like you'd expect. Really allosteric, powerful activator of the PFK reaction. At the same time, there's this other molecule here, fructose 2,6-bisphosphate. You recognize that? We've seen fructose 1,6-bisphosphate. Where'd fructose 2,6-bisphosphate come from? That just basically came out of left field. We haven't ever talked about it before. This is basically fructose, but not with phosphates symmetrically on the 1 and the 6 carbon, but 1 on the 6 and 1 on the 2 alcohol group. And it is an extremely powerful allosteric activator of liver PFK. And to understand why, we have to go back and think again about this idea of blood glucose physiology. But before we do that, I want to show you a little bit of the structure of PFK, because I keep talking about allosteric enzymes, but I haven't really shown you an example of one. There are a bunch of them. We could do a bunch. The basic ideas, to a first approximation, fall in line with what we talked about with respect to hemoglobin. But just to show you an example, here's a structure of PFK. And I can't remember if this structure is liver PFK or muscle PFK. There are some differences between them in some very important ways. But to a first approximation, this gives you an idea, right? It's a homotetramer, 35 to 40 kilodalton monomers. Here's one subunit here in this sort of light blue, you can see. Here's another one in the dark blue, and they make sort of a homodimer. And you take two more of those and you put them on the backside, and you've got the PFK fundamental tetramer. There's some evidence that PFK actually may function in larger complexes than this, but this is the fundamental enzyme. And the active site, you can see, is right here, sort of along this edge, right, of one monomer. There's the ATP, right? There's the fructose 6-phosphate. You got a symmetric active site over here on the other subunit. And the activity of these active sites, all four of them, are linked up both in terms of an allosteric response to ATP levels, into ADP levels and citrate and all these other things, substantially by a set of regulatory sites that exist in this cleft at the top and the bottom of the tetramer, right? And what happens is, you can think about it, at least to a first simple approximation, again, like hemoglobin, as sort of a TR type equilibrium at the highest level of tetramer structural change. It's more sophisticated than this, right? Like we saw with hemoglobin, which is one of the simplest ones. It's pretty clear that this model alone really is not sufficient to fully explain at all what's going on with an enzyme as elaborately regulated as PFK. And if you want to learn more about that, I'd encourage you to talk to Dr. Reinhardt, because this enzyme was one of his babies. And he's really dissected the allostery of this enzyme in great detail. But for our purposes, to a first approximation, we can think about it as sort of a structural shift between two sort of macro states of the tetramer. And that shift occurs, or it's a big part of the allostery in regulating this enzyme, occurs a structural reconfiguration, kind of like we saw with hemoglobin, where the dimers sort of rotate into each other, right? They sort of iris towards each other, and that irising in towards each other impacts the activity of the four active sites around the enzyme. And again, it's not at all a simple, clean MWC-like transition. It's considerably more sophisticated than that, but that's one important level at which the structural changes of the overall oligomer are affecting, in response to allosteric effectors as well as substrate, are affecting the activity of this enzyme. Okay, so just to sort of give you a little bit more of what this might look like, here's another view of this enzyme. Here's those active sites in the tetramers. Here are a couple, I think this is ADP, or it might be ATP in one of the effector sites. I can't remember, up at the top of the tetramer. And to give you a sense of what this looks like, here again is the response of PFK in the absence of anything else, responding, or any effectors responding to one of its substrates, fructose 6-phosphate, and it responds with a classic positive homotropic response to PFK. It's pretty cooperative. You can see there's the initial velocity. There's that nonlinear curve, and it's responding to its substrate, PFK, as a nonlinear positive homotropic response, right? Great. It also responds to ATP in an interesting way. ATP is a substrate, right? So you put in more substrate, the enzyme is going to go faster, but it doesn't respond, it's going to make more product. You put in more substrate, as we see with any enzyme, it's going to make more product. So here, if you look at the addition of ATP, you add ATP as a substrate, the activity goes up and up and up. But instead of doing sort of like, classical Michaelis-Menten or even responding allosterically, what happens when you get to a certain level, at the ATP concentration, the activity collapses, right? That's that very powerful sort of negative allosteric response to ATP levels that is mediated by ATP binding to one of these regulatory sites up here at the top. So that's a really interesting kind of response, right? It's not product inhibition, because this is actually a substrate. It's the substrate also binding to a second site on the enzyme as a very powerful negative sort of non-competitive inhibitor of the activity of the enzyme and through an allosteric response in these other sites. All right, but you'll also notice something else on these plots. There's that fructose 2,6-bisphosphate that I talked about, right? Look what it does. Fructose 2,6-bisphosphate is a massively powerful, positive allosteric effector for the output of this enzyme, dramatically pushing PFK into a much more active form, right? At much lower substrate concentrations of fructose 6-phosphate, right? Incredible stimulation. At the same time, fructose 2,6-bisphosphate can dramatically, not totally, I mean, ATP levels are still dropping this guy's activity down, but it can overwhelm the activity or the inhibitory activity of high concentrations of ATP if fructose 2,6-bisphosphate's around. So it's a very powerful activator of liver PFK. And the interesting thing is, this allosteric effector and the enzyme that makes it, you would think this, metabolism is so old, it's been worked out. It was only discovered about 20 years ago, maybe a little less than 20 years ago. So this is a relatively recent addition to our understanding of how a really core piece of basic metabolic physiology works. To understand that, I need to go back to that idea of the cyclic cascade and elaborate it a little bit. Here again is that cyclic cascade. I've talked about that post-translational modification of an enzyme by a post-translational event, phosphorylation through a kinase, and then of course the attenuation through a phosphatase. And we talked about how we have a kinase enzyme that then once it's activated can activate the target enzyme here on the phosphatase. Same thing, once it's activated, can deactivate it. But now let's elaborate this one a little bit more. What if it turns out that kinase was itself susceptible to a post-translational activating event, right? In this case, the kinase is activated by phosphorylation, right? A kinase that then comes in response to some effector, right? That effector response now activates a secondary kinase that comes in, activates our primary kinase, which can now come in and activate our target enzyme. If you think about the fact that the way we talked about a cyclic cascade before, that each of those activated enzymes here can have it's sort of a force multiplier on what you can do with the target enzyme, because each of these guys is an enzyme, it can hit multiple of the target enzymes, right? So that's a big increase, amplification in response. Well, now we've doubled down on that, because now we've made that enzyme now a responsive to another effector, right? So the actual effector now here, very small amounts of this effector can do something, be sensed by this primary kinase. Each one of these guys will now hit a whole bunch of the secondary kinase. Each one of those can now go hit a whole bunch of these guys, right? So now you can imagine this idea of stringing a whole set of these sort of amplifying post-translational modification events together. And hopefully the idea now would be that, or you could see, you can get enormous signal amplification, not just 10-fold or 100-fold, millions of fold, maybe even more. You can get sensitivity so sensitive that you can measure just a few of an initial effector event when you string a whole bunch of these kinase cascades or post-translational enzyme cascades together, because each stage massively amplifies the events of the previous stage, because each output is an enzyme that then spreads out, hits lots of targets, each of those spread out, right? So it's a huge signal amplifier. Okay, and we can make this even more responsive in some ways by having these effectors, either the kinase that deactivates the phosphatase that deactivates the primary kinase, or the secondary kinase, or the primary kinase. Either one of these guys themselves now responding to their effectors here allosterically, we can now get incredibly exquisitely sensitive responses to changes in our presence of that allosteric effector in terms of the output of this final enzyme, right? Okay. All of that is sort of set up to sort of explain how fructose 2,6-bisphosphate and how coordinate regulation of all this stuff is kind of coming together. And I sort of have to go back to some basics of blood sugar physiology that is hopefully a reminder for you guys. You guys all remember, right, that blood glucose level is a big deal. We've talked about why that's a big deal. And hopefully you remember that how that's regulated in organisms like us. There's a key organ, of course, the pancreas, that's sensing blood glucose levels. This is one of the main things the pancreas is taking care of. And as blood glucose levels change, it synthesizes a couple of small hormones, insulin in one case and glucagon in another case. When blood glucose levels get high, pancreas senses that, stops making glucagon and starts making insulin. So when blood glucose levels go up, insulin levels go up. That insulin is a target, multiple targets, but a big one is the liver, right? And what it does in the liver is it synthesizes the storage and conversion of glucose into other things. Basically, blood glucose is too high. The liver needs to suck that glucose out of the bloodstream and store it, all right, in one of a couple of ways, right? One, of course, is it upregulates glycogen synthesis. It makes glycogen granules and it packs up its glycogen storage locally. The other thing it does when insulin activates hepatic tissue is it activates glycolysis, right? And that glycolytic flux turns some of that glucose into acetyl-CoA, which is then run through fatty acid biosynthesis to make fatty acids, right? And so all of that results in a removal of glucose from the bloodstream, storage in glycogen, and storage in fatty acids, right? And blood glucose levels can then be pulled down. Now, blood glucose levels get low, right? Maybe the system overshoots or you haven't eaten in a while. Under those circumstances, pancreas senses the low blood glucose level, stops making insulin, and starts making glucagon. And glucagon has the opposite effect. Glucagon hits liver tissue. It stimulates the liver to take those glycogen stores, break them down, turn them into glucose, and export that glucose into the bloodstream for the peripheral tissues, right? Glucagon also shuts off glycolysis, because if the body needs glucose, the last thing you want to be doing is making fatty acids, right? So it shuts off the conversion of glucose into acetyl-CoA, shuts glycolysis down, upregulates gluconeogenesis, and you start making glucose either from glycogen stores or other, those 3 or 4 carbon stores we talked about before, lactate, for example, amino acids, and you make glucose and export it into the body. Okay, that's the physiologic picture. How does this fit into what's going on with these pathways? All right, so here's fructose 2,6-bisphosphate again. Remember, it's fructose, but instead of the sugar, instead of the phosphate on the 1 sugar, it's now on the 2 sugar, which is the anomeric carbon in fructose, as you recall. This enzyme that makes this is, of course, known as phosphofructokinase, but PFK2. All right. Again, enzyme was only discovered, I think, less than 2 decades ago. Right. It's a tiny enzyme compared to PFK1, and it's a bifunctional enzyme. So it's got a kinase domain and it's got a phosphatase domain. The kinase domain will take fructose 6-phosphate and make fructose 2,6 by putting that phosphate group on. The phosphatase domain will then take that off and convert fructose 2,6-bisphosphate back into fructose 6-phosphate. So how does this all work? All right, well, that small ligand effector, fructose 2,6-bisphosphate, as I said before, is a powerful regulator, enhancer of fructose, of PFK, phosphofructokinase. It's also a powerful inhibitor of the opposing gluconeogenic enzyme at its committed step of fructose 1,6-bisphosphatase. All right, so the way this is thought to work is in the following fashion. When blood glucose levels are high, right? The liver's blood glucose is high, insulin's up, the liver's taking all that glucose, it's shoving it into glycogen, it's trying to pull blood glucose levels down. It wants to burn off that glucose, turn it into acetyl-CoA because we're super abundant, make fatty acids, right? Under those circumstances, the kinase domain of PFK2 is active, taking ATP, it's taking fructose 6-phosphate, which is now abundant, and it's turning it into fructose 2,6-bisphosphate, which supercharges PFK, because you want to run that glycolytic flux, you want to burn off this glucose. You want to burn off the glucose that you're not storing into glycogen, you want to turn it into acetyl-CoA, you make fatty acids out of it. Okay, but now what happens when blood glucose levels equalize, right? Now you've burned that stuff all off. Now, blood glucose is going low. Pancreas stops making insulin, makes glucagon. The glucagon then comes in, interacts with a G-coupled receptor in the receiving liver tissue. And that activates, like I said, small G protein, which activates an adenylate cyclase, right? So first stage of our cyclic cascade is an enzyme, membrane-embedded enzyme, that takes ATP and makes cyclic AMP. And that cyclic AMP is a powerful effector that then turns around and hits protein kinase A. Protein kinase A phosphorylates PFK2, and that phosphorylation event shuts off the kinase domain and activates the phosphatase domain. What this does is now shifts PFK2 from making fructose 2,6 bisphosphate to now turning that fructose 2,6 bisphosphate back into fructose 6 phosphate. So when that happens, you shut down PFK dramatically downshifts, right? Because this powerful allosteric effector decreases dramatically. This reduces the glycolytic flux, which makes sense because now the body's screaming, I need glucose, I need glucose. Stop turning it into fatty acids. You want to stop that glycolytic flux, let the liver now break its glycogen stores back down, run gluconeogenesis, and export that glucose out into the peripheral tissues that need it, right? At the same time, there's a kinase, there's a protein phosphatase that will complete our cyclic cycle here on PFK2, that will then clip this guy off and return the PFK2 to its most active form. But it responds to fructose 6 phosphate as a positive activator, which makes sense because if you've downregulated PFK2, right, and now blood glucose levels start going back up, right, fructose 6 phosphate pool starts to build up because the enzymes coming in are starting to make fructose 6 phosphate. This enzyme will now say, uh-oh, we got plenty of glucose around, the insulin levels are going back up. We now need to reactivate PFK2, push it back over to the active kinase, remake fructose 2,6 bisphosphate, and off we go, right? Reversing that. Okay. So that is sort of a first order picture of how aspects of these different pathways for glycolysis are regulated. And sort of in a final big picture view, so much of what we've been talking about is focused on glucose, right? And you're going to see how those acetyl-CoA equivalents, like I mentioned, are going to eventually can be turned into fatty acids, right? And so the flux down this pathway glycolytically to make pyruvate is a really big deal, right? And we've seen how there's an opposing flux running through gluconeogenesis that can take things that can be turned into pyruvate, right, and move it back into glucose, right? Essentially by using oxaloacetate to recreate phosphoenol pyruvate, right? Not so directly through pyruvate. And we've got these two opposing set of enzymes that control the beginning or end of these pathways. For glycolysis, it's hexokinase, phosphofructokinase. At the end, right, it's pyruvate kinase. And then for gluconeogenesis, it's PepCK, right? And then the two phosphatases up at the top of the pathway that then turn fructose 1,6 bisphosphate back into glucose, right? And these guys are turned on and off depending upon which pathway you want to run. When these are on, these guys are off. And when gluconeogenesis is on, glycolysis is off, right? And now we've seen that there's an additional enzyme regulated in the same way, right? The effects of PFK on these guys are opposite. The effects of fructose 2,6 bisphosphate on the gluconeogenic enzymes are opposite what they are on the glycolytic enzymes, right? Fructose 2,6 bisphosphate is a powerful allosteric inhibitor of some of these enzymes, while it's an activator of the glycolytic enzymes, right? And then there are also feedbacks up into the formation and storage of glycogen, which we've really barely just touched on in terms of the breakdown and synthesis that are also very carefully coordinated. Okay, so with that, that more or less covers what I want to talk about here from the point of view of the specific pathways. Now, the last thing I'm going to hit on in the final video is sort of a consideration of sort of a bigger picture of the behavior of these networks.